Okay, so let's start with our stem cell, our theme of the stem cell. Actually, I should not share this slide with you guys, but I think that is fine for you to dig and to explore these timelines with respect to the presence and absence of certain critical proteins which are involved in the process of differentiation. So when everything starts, the cells present within the embryo, each one of that cell is capable of doing wonderful things, is capable of becoming everything. But over the period of time, they started becoming different and different. At one stage, they are called as totipotent. Totipotent cell is they can be everything. Somehow the stem cells are also totipotent, but traditionally the term totipotent is used for an earlier versions of stem cells, like a better version of stem cells, even the stem cells who have the highest potential. But what we studied that even at the stage of late blastocyst, when we have a cavity and when we have an inner cell mass, this thing is called as inner cell mass. All of these cells from 100 or more cells are capable of producing everything. Each one of that cell has a complete potential to become a baby. Each one of these cells is able to become a baby. The underlying regulators which are controlling these different phenotypes are complex. I'm not telling you that this is only one or two genes which are responsible in these transformations, but I want you to allow your brain and allow your wisdom to think and ponder on these complex activities. I'm just giving one of the example here with this slide, that in case of OCT4, it's a kind of a master regulator of this process. But once again, I don't want you to believe that this is the only thing. There are other factors which was written here as well, FGF4 or fibroblast growth factors, which are involved, that how these cells will going to interact with each other, and also how some of the cells will become like an outer layer. So these little differences will ultimately create, you can see, things. One of them will become like an epiblast. Epiblast is like an outer cells, what you can say. And some of them will create a different type of cells. So during the process of development, what we observe, a small change at the start will ultimately create a very big change in the phenotype of the cells after a few days. And that is because of the increase in some of the regulators or some of the master regulators which influence the expression of downstream target genes. One of the example is OCT4, but there are others as well. So in totality, in a summary, the embryonic stem cells from the inner cell mass, from there, these are the inner cell mass. We extract cells from the inner cell mass, we culture them in the laboratory, and we can differentiate them into different types of cells. Almost all possible types of specialized cells can be created out of the embryonic stem cells. Those embryonic stem cells are also called as pluripotent. They have the potential to become everything. The embryonic stem cells, dealing with the embryonic stem cells is has a lot of challenges. Some of the challenges I have shared with you guys earlier, that it depends mainly on the availability of the egg. which is a very precious commodity, because the sperm and egg, they will going to create the zygote, and that zygote will going to develop an early embryo, and that embryo will going to give you what? That will give you the embryonic stem cells. So whether we like or not, whether we accept or not, the embryonic stem cells comes from a life, comes from an embryo or a potential baby. So that's the first thing, that there is not the supply of egg is not unlimited. That's a very big limitation. As long as we are not having as long as we are not having the as long as we are not having unlimited supply of we are not having unlimited supply of eggs. It's a precious commodity, so we cannot expect an unlimited supply of embryonic stem cells as well. So somehow, we like or not, it depends on the availability of eggs, and this is a limiting factor in embryonic stem cell research. So when people have discovered this thing, they thought that they have found a way to create all sorts of cells. All they need in embryonic stem cells, let them grow in a condition A, it will become like skin. Let them grow in a condition called B, and they will be like neurons. Let them grow in a condition called C. I will be asking about these three conditions, okay? I might be asking you about the conditions. Please search it, and they will become liver. So that seems very easy, but the problem is the supply chain. We don't have embryonic stem cells that much. So that actually the bottleneck of the research associated with embryonic stem cells. And it also raises lots of ethical concerns as well. Because who will going to donate the egg, who will going to donate the sperm, and what will happen to that embryo, whether we should consider that embryo as baby or not, whether that thing has some sort of rights or they don't have any right. So it's quite complicated, I can tell you. To make things a little bit easier, this complicated slide might help you to categorize these things into different categories. I want you guys to work on these individual categories, like work on these individual boxes. I don't know, no wonder these might be your future area of research. So this will going to help you like a primary slide to feel that what type of expertise or things you need to know about your area of research. For example, everything starts from the early embryo, inner cell mass. You can take out those cells, and those are called as pluripotent embryonic stem cells, most powerful cells till today present on the planet Earth. And they can become everything. It depends on the path that what will be the destination of the embryonic stem cells. They will become like this, mesodermal stem cells. Then they can be like ectodermal stem cells. They can be like an ectodermal stem cells or mesodermal stem cells. The mesodermal stem cells might be like, if you can see, hematopoietic stem cells. And hematopoietic stem cells might be able to produce a lot of other types of downstream cells. The journey might be different. The mesodermal cells becomes like endodermal cells, ectodermal cells. These are the primary categories we have studied: mesoderm, ectoderm, and endoderm. The endoderm give rise to lungs and the digestive system. The endoderm will give rise to lungs and the other things. The ectoderm will going to give rise to the skin and the brain and the spinal cord and eyes and very complicated functions and things. So every complex organ arises from preliminary cells. All those preliminary cells come from the pluripotent stem cells. So what I can tell you in a summary, that the pluripotent stem cell originally select to travel in three different directions. One of the direction is called as endodermal stem cell. That stem cell is capable of producing all the things associated with endoderm. But that stem cell will not be able to produce anything which is associated with mesoderm or which is associated with ectoderm. So the potential of this stem cell is limited. If the potential of this stem cell is 100, this might be like 95. Oh, sorry. And this will become less and less and less. So the potential of the stem cells is now reduced because it has a defined destiny to produce the endodermal related things. Sonani, please, I'm really sorry, okay? Thodi se jagte rehna. From Monday, I will be able to teach you guys on camera, okay? I'm sorry, bas thoda sa boring hai. And similarly, when the cell choose to enter into the destiny or into the path called ectodermal stem cells, it will only be able to produce these things. So one way or another, when a cell choose to commit a certain path, the level of differentiation goes up and the level of stemness goes down. And cells ability will be limited. If you ask me, sir, this cell is pluripotent stem cell, the answer is no. This is not a pluripotent stem cell anymore because it will not be able to produce everything. If you ask me if this cell is a pluripotent stem cell, the answer is no. It is no longer a pluripotent stem cell. So you will be able to see that what is the timeline as well as the steps to differentiate into different types of cells present in human embryo. And that is a quite, I must say, informative slide for you guys. Okay, please stop me if you have any question, okay, without any problem. There are two types of stem cells. The one you have just studied, the embryonic stem cells. There are other types of stem cells which are present in the adults. They are called as tissue stem cells, or they are also called as adult stem cells. They are present in brain, in the eyes, in the skin, in breast, intestine, testicles, and muscles, in bone marrow especially. All these places are the places where you need a constant supply of cells. You will see all these places are the ones where you need a consistent supply of cells, consistent supply of new cells. Think about the bone marrow. The job of the bone marrow is to produce all types of blood cells, T cells, beta cells, red blood cells, platelets, all are produced here. There are some primary precursor cells which are present in the bone marrow, and these primary precursor cells are responsible for the production of all of these different types of blood cells. They are called as hematopoietic stem cells. They are called as hematopoietic stem cells. The hematopoietic stem cells are present in the tissues. They are not like the pluripotent stem cells. They are not the ones which can produce everything. They are the ones which can produce only the blood cells. So they are called as tissue stem cells or adult stem cells. Or sometimes the more accurate name is a multipotent stem cells. So the multipotent stem cells mean the stem cells which are capable of producing a certain category of cells. There is a huge potential out there in the research of the tissue stem cells as well. You can get the tissue stem cells related to the blood which are found in the bone marrow, as I was talking about, hematopoietic stem cells, which are responsible to produce all these types. All these different types of cells are coming from one precursor or one original cells. Many of the blood diseases, especially many of the blood cancers, they arises due to the error in this cell. They arises due to the error in this stem cell, which are present in the bone marrow. So if you able to get those cells, and if you able to engineer those cells, to fix those cells, made the corrections and put back into the bone marrow, you might be able to fix many of the types of diseases, of blood diseases. They are multipotent in nature. So the difference between the adult stem cells and the embryonic stem cell is that the adult cells, they are not capable of producing everything. This slide might be easier for the people who studied the immune system, but for the other people, just focus. It's not that much. It's not that much difficult. Our bone marrow contains hematopoietic stem cells. This one, it's not a stem cell. It's a multipotent stem cell. When I said multipotent, it is only capable of producing a certain category or certain types of cells. Over the period of time, when it gets differentiated, it produces a precursor cell. This is a precursor cell, which is capable of producing this thing and this thing. Now, this thing is a different journey. The name of this cell is lymphoid progenitor cells. Means they will going to produce the cells of the lymphatic system, or they will going to produce the cells of the immune system, or they will going to produce the T cells, if you remember, or they will going to produce the beta cells, they will going to produce the natural killer cells and other cells. So when the process of differentiation occurs, certain genes the expression goes up, the other ones goes down. Because of the difference in the patterns of expression, the cells choose a different track. And this different track will take that cell to a different outcome or a different journey. In other case, the cell will proceed in this myeloid precursor cell in a different patterns of expression. And then they will going to produce neutrophils and different types of white blood cells. You can see eosinophils and other monocytes and the red blood cells. Most cases of blood cancers occur because the cells lose the control on the pattern of expression somewhere here. Can you see there must be a tight balance between the patterns of expression of different genes? Otherwise, the cell will going to choose a different direction on a different destination. So if anything bad happens in the control of expression, what will happen? The cell will choose a different pathway. In most cases of blood cancers, what happens? The cell is not able to control the pattern of expression in these stages. As a result, you might remember the people with leukemia, they will develop abnormal proportion of blood cells. Sometimes they do have, lot of white blood cells as compared to the red cells. Sometimes they are overproducing the T cells, different types of lymphomas, the cancers of the immune system, it comes from there. So that is how we can categorize the adult stem cells. Understanding about this mechanism, what genes are supposed to be activated, which turn and change the destination of the cells from here. What type of genes are required to be expressed in what proportion, which pushes the cells to move from here to there, and then from here to there, from there to red blood cells. All these information is very critical for our future endeavors, because we want to create cells of our own choice. And that is only possible if we understand how nature produced these different type of cells out of a common precursor cell. The beauty and potential is the presence of a common precursor. This is the beauty, that we have a start point. We have one cell. All we need to know is how to tune this one cell to create all these beautiful products of life. So I believe in the future, when we have more information about the patterns and the changes in the patterns of expression of the genes, we develop more techniques to control these patterns. We will be able to have more control on producing these different types of cells by our own will and wish. It's a huge, wonderful area of research. So to get the things, to understand things in an easier way, embryonic stem cells, cells from the inner cell mass, they are able to produce everything. You can culture them, you can grow them, and they will going to become like everything. You can convert them into the nerve cells or the liver cells. On the other hand, the adult stem cells, which are coming from the bone marrow, you can culture them, you can deal with them, but they will only be able to become few types of cells. Like the bone marrow cells will be able to produce the blood cells, T cells or red blood cells or W, but they are not able to produce everything. That was the limitation. So people are using these things in different types of medical areas called as regenerative medicine, using embryonic stem cells and regenerating important cells and repairing the damaged cells, like producing neurons. The neurons which are able to replace the faulty neurons, and trying to fix the Parkinson disease. We are producing other using human stem cells to produce cardiac cells to replace the cardiac muscles, to avoid cardiac failures. This is a very critical stuff. This is very exciting for me as well. Pancreatic cells. Engineering human embryonic stem cells, pushing them to differentiate into pancreatic cells. Because the pancreatic cells, their job is to produce insulin. And most of the diabetic patients, they are having trouble because they are not able to produce enough insulin. Their pancreatic cells are not working in a normal way. So we can replace the pancreas. I believe the nearest possible reality is that we might be able to produce liver cells or the entire liver, or maybe the pancreatic cells, or might be the pancreas or bone marrow cells, because they are not organ in that way. They don't have defined boundaries. Liver is the most easiest of the thing, I believe. It does not have a very well-defined shape or organization. So we can produce liver cells, we can produce a collection of cells which can execute the function of liver. That might be the most nearest possibility, I believe, and the pancreatic cells. And we can create bone marrow cells and inject to fix the leukemia and other things. So these are very wonderful, exciting options with the help of stem cells that are out there. Now the problem is that we are dependent on this precious commodity. I was talking about these things again and again. The precious commodity, the egg. And also, this is the precious commodity, and also the ethical concern. The ethical concern of dealing human embryo. Whether we like it or not, the human embryo is like a human life. So in order to deal with this ethical concern, because this is the point and a concern of people, that you are utilizing human embryo for research. There was a need, because this is a bottleneck of research, embryonic stem cell research. There was a need to find an alternative routes, alternative pathway to have stem cells. So what scientists have developed at that time, that what we call is known as iPS, or induced pluripotency. So let me talk this thing in detail, and then I will come back to the slide. The concept of induced pluripotency or induced pluripotent stem cell is that when we have a differentiated cells like the skin cell, when we have a skin cell, it's a future, it's a differentiated version of a cell. We can reprogram the expression, we can reprogram the genes of that cell, and we can convert them back into a pluripotent stem cells. Now this is not the embryonic stem cell, but it behaves like an embryonic stem cells. This is not by default embryonic stem cell, but we push and change the expression pattern of a skin cell and push them to start behaving like an embryonic stem cells. So such a stem cell is called as induced pluripotent stem cells. It's very fascinating. It was like a big breakthrough of the century at that time. We thought we have discovered the master key, that we know how to convert the skin cell, which is the future. If you remember my earlier talk, it's like a future. And embryonic stem cell is like a past. So we convert future back into the past. The embryonic stem cells, they are capable of producing all sorts of cells. The induced pluripotent cells, they are also able to produce all sorts of cells in the same way. And that was quite exciting, that the embryonic stem cells like the embryonic stem cells, the induced pluripotent stem cells also are capable of producing different types of differentiated cells. Think about getting a cell, a skin cell, converting them into stem cell, and then producing a neuron. From the skin cell to the neuron, from the skin cell to the red blood cell, from the skin cell to the other type of cells. That's very fascinating. Please
sir,
ask your question
when we
on screen, okay? Please ask your questions on. That is much easier. Okay, जब तक कोई sensitive सवाल ना हो जो आप सबके सामने नहीं पूछ सकते हैं, then I will go to the private messages. Okay? जी, शाबाश.
Sir, when we are de-differentiating a cell and then we're differentiating them again, does it have any impact on the stability of the genome? Because we're doing a lot of things to those cells.
Yes, of course.
There are changes.
There is a paper I suggest you to study, but there are a lot of paper out on this question. You know, this is the what your question is that. Let me simplify your question and let me tell you what you need to do after that. Okay. So this is an iPS. What that means? That this is an embryonic stem cell produced out of the skin. Okay. Now this one is stem cell, embryonic stem cell. If I ask you if this is capable of producing the baby, the answer is yes, perfectly. If this thing is capable of producing the baby, the answer is yes, perfectly. So now what we do know is that we believe that these two things are equal. Am I right? Zunaira, are you with me?
Yes, sir.
Believe that these two things are equal. Because whatever that stuff is capable of doing, the other thing is doing the same thing. So मुझे लगता है ये दोनों एक ही हैं. But people have studied. लोगों ने क्या किया? लोगों ने कहा कि यार, of course these are two different routes. It's pretty natural. Like a safe, secure, a very hygienic way of producing an embryonic stem. Now this is like laboratory में rigorous, doing things, changing expression pattern, giving things. So people were wondering about, is there a difference between these two things? So what they have done, they have analyzed all the patterns, all the expression of all the genes here, and then expression of all the genes here. I suggest you to go and see that paper, similar sort of papers out there. Then they compare the patterns of expression of genes between these two. तब उन्होंने compare किया that what type of genes are expressing here, what type of genes are expressing here, what type of genes are expressing more here, what type of genes are expressing less here. They compare the expression of all the genes in these two states. And they came to know that there is hell of the difference in the patterns of expression of these two. Oh my God. So, although we are able to produce a baby, although the induced pluripotent stem cell is capable of giving us the baby, like the embryonic stem cell, still we don't know everything. That is an indication that still we need to know much. We need to know much more, that what are the defining factors who are allowing this induced pluripotent stem cells to produce a baby, and why still there is a lot of difference between these two. So those who are interested in stem cell research, they should go and see the difference in the patterns of expression of these two types of cells. They came to know, they will be able to understand that still there is lots of area which we have no answer, that even then we are not able to rationalize the difference. We don't know why the difference is there, but somehow this induced pluripotent cell is able to produce all the possible cells. So that means that we came to know a phenomena, but we don't know all the factors behind this phenomena. So ये वो question है. This is the question which must be answered before this technique will be used in the future to clone humans or to clone the babies. Because if we produce a baby out of the skin cell, and the pattern of expression is not similar to the embryonic stem cells, who knows, the baby will going to develop something very different in the far distant future. So scientists are still working to know and to discover big difference between these two things. Okay, so I also जी शाबाश पूछना.
If they actually make clones through that, isn't it like dodging the ethical concerns if they begin making clones of humans using this?
Yeah, that's a different topic. I mean, whether we should do this or not, but scientifically speaking, think as a researcher, if this will be your task, if somebody give you a task, or if you choose to create a clone, तो आपको सबसे पहले क्या करना होगा? You need to be very careful, and you need to know everything about the induced pluripotent stem cell. तो दो काम करने पड़ेंगे आपको. If you are interested in executing this research, दो काम करने पड़ेंगे. First, you need to know embryonic stem cells के pattern क्या होते हैं. You should know everything about the embryonic stem cells, and then you should know everything about the induced pluripotent, because you will going to use this one to clone the humans. So as a researcher, you are interested in knowing that although the induced pluripotent stem cells are producing everything, but still they are not similar to the embryonic stem cells. Is that difference matters or not? Now, this is a separate question, the whether we should go for human clones or not. Okay? हमें इसको address करना चाहिए, हमें इस पे बात करनी चाहिए, and we will talk on these things. But the first thing is that as a researcher, if you want to go for human clones, you want to know everything about the induced pluripotent stem cells, so that you might be able to answer all the wonderful things, all the special things which will going to happen, which will going to unfold in the distant future. तो क्या आप बता सकते हो कि ऐसा तो नहीं है कि आप स्किन सेल से डिफरेंशिएट करके you will start producing the neurons, and that neurons will stop working after 10 years. That neurons will start producing something very bad in the brain of the patient. So we should know about the answers of all these things. Okay? I suggest you, if anybody wants to talk, please feel free. I suggest you to read this thing. And this is the first paper, I believe, where we found that a skin cell or a fibroblast cell is converted into a stem cell. And that's very fascinating study of that time. And that we came to know that is because of certain factors, because of the OCT3, because of the OCT4, because of the SOX2, because of the KLF4 and c-MYC. These are the 4 master regulators which were discovered, and these 4 alone are enough to transform this fibroblast into a stem cell. And you know the ultimate test of the stem cell? What is the ultimate test of the stem cell? Is to put it into the womb of a surrogate mom and to see whether it can produce a healthy baby. If it is produce a healthy baby, then this thing can be considered as equally strong, equally potent as embryonic stem cell. But please remember, this is not coming from the sperm and egg, it is coming from the fibroblast because of these 4 wonderful master regulators. And these 4 were given with the help of the retroviruses. In mouse, it was done in 2006. In humans, it was done in 2007. Of course, after creating the human embryonic stem cells, they did not use this to position. So in mouse, in 2006, it was done, and they produced the mouse babies as well. But in case of humans, they have created the embryonic stem cells. Of course, it was not reported that they have produced any babies, human babies, but who knows? There are always villains out there, you know, the scientific community as well. But the big question was addressed. If the embryonic stem cells, they have the potential to fix a lot of things. Recall this slide. Sorry. I'm sorry. Let me go back. Recall the slide which shows you the potential of stem cells. If the stem cells have that much potential, like this one, if the stem cell is like doing all these wonderful things, you must be interested to find lots of human embryonic stem cells. Now, human embryonic stem cells is not nice to get from the human egg. It's not nice to get from the human embryo. It seems very scary. It's weird. It's unethical. So how about producing pluripotent stem cells out of the skin or out of the differentiated cells? And that was a discovery, and that was special, and it gives these people a Nobel Prize out of it. These 4 master regulators, they proved to be good enough. I don't know why only these 4 are good enough. But they have found that only these 4 are capable of transforming a fibroblast into a pluripotent stem cells. And in this case, it is called induced pluripotent stem. Now, this is capable. You can do 2 things from here. The one thing, you can produce a baby. The other, you can produce the tissues. And that's what we are doing, producing tissues, neurons, cardiac tissues, other tissues to fix the damage. If you ask me, sir, how it is possible that these 4 factors are controlling and regulating everything, I can tell you the landscape. And I want you guys to wander in this valley to understand the details. These 4 factors like SOX2, OCT4, more accurately OCT4, but OCT3 is there, KLF4 and CMYC. They are the master regulators. They have different target genes. Luckily, the genes they are targeting, that's a very interesting question. The genes they are targeting, they are themselves master regulators. They are not controlling 1 or 2 genes. They are controlling cascades. They are controlling series of events. They are controlling several important transcription factors. So when the 4 of them were expressed in a cell, in a fibroblast cell, they activated the target genes. That's the first thing. Most of the target genes, they produce transcription factors, means they are producing proteins. The transcription factors are the proteins which, when bind with the promoter region, of a gene, they push the genes to produce the messenger RNA, RNA pol 2, with the help of RNA pol 2, or they push the cells to produce proteins. So because of those transcription factors, those new transcription factors, we will be able to have new messenger RNA, new proteins, means we will have new functions. And that new function will going to transform and change the phenotype of the cell. But this is not enough. This is very small side of the picture, a very small thing of the picture. Having transcription factors is not good enough. Actually, many of the transcription factors, they are epigenetic modifiers. I wish I can spend more time on this thing. The chromatin topology, how our genes and chromosome is present, like in this way, or they are present in this way. How our genes or chromosome is oriented within the nucleus. It defines what type of genes will be expressed. There is an order in this chaos. Just let me try to explain this thing in an easier way. So let's suppose we have a chromosome here. It has this side folded like this, and we have this side opened like this. So all the genes which are present here, they are less accessible, and all the genes which are present here, they are more accessible. So they are supposed to be expressed more easily as compared to this one. All the genes present here, they are less expressive, they are more expressive. So the patterns of expression of this cell will have expression of 1, 2, and 3, and not 4, 5, and 6. What these things are doing, they are changing the topology of the chromosome. They are changing the epigenetic markers, which actually defines how this chromosome will be present in 3-dimensional time and space. The chromatin topology of this gene is changed. For example, it will be like this one, a change. So the genes like 1, 2, and 3 is now present in an inactive domain, while the genes of 4, 5, and 6 is now present in an open topology, an open conformation. So the same cell which is earlier producing 1, 2, and 3 and not producing 4, 5, and 6 is now producing, now not producing 1, 2, and 3 and now producing 4, 5, and 6. The pattern of expression is changed. And you know, it's not just changing the pattern of expression of one gene. The chromatin topology is able to change the pattern of expression of many genes in one go. And that will going to change the fate of the cell. And that is very critical and special. Certain other things as a researcher you should keep in your head when you are working with the stem cells, and especially the induced pluripotent stem cells. For example, if you have a somatic cell, epithelial cell, and you send OCT4, SOX2, KLF4, CMYC, what these things are supposed to do? These things are supposed to change the dynamics of the cells. They are called as Yamanaka factors because Yamanaka has discovered it, awarded with the Nobel Prize as well. What the cells are doing. So having all these things will push the cells to go for to become pluripotent. But remember, our cells has a natural defense mechanism as well. And one of the most famous of them is the P53 pathway. Anything weird when happens within the cell, it triggers the P53 pathway. When people were trying to put OCT4 and these Yamanaka factors to convert the somatic cell into a stem cell, the P53 pathway is like an enemy. Here, it's like a resistant force because these 4 factors will change the dynamics of the cells a lot like this. I mean, it's like a chaos. It's like a big change. It's like you are transforming Earth to become like Mars. It's like changing the dynamics of the cells that much that the cells will started thinking something very weird is happening. It activated the P53. And once the P53 is activated, usually the P53 is supposed to get activated in an oncogenic situation when the DNA damage is there. So a weird sort of activity will push the P53 to get activated, and it does 2 things. The first, it actually arrests the cells to proceed to go further for the cell division and try to repair the damage, and then it will going to go for the apoptosis. One of the factor KLF4 here, the KLF4, the KLF4 is actually block the P53 for the activation. So the other things can execute the job. If the P53 is not there, the Yamanaka factors will eventually able to push the cells to become induced pluripotent stem cells. So the people who are working and researching and producing induced pluripotent stem cells, they need to suppress the P53 in the meanwhile. They need to find a way. They can find a way using small interfering RNA or other ways. They need to find a way how to block the P53. So the P53 should not get activated and should not feel something weird is going on. As long as the cell is not feeling something weird is going on, the cell will slowly transform themselves into an induced pluripotent stem cells. And that is very interesting as well. I wish you guys should be able to go through all these things. So a lot of options are out there. Reprogramming human cells, converting them into stem cells, in vitro differentiation, producing whatever you like to, neurons, novel drug targets. You can identify a lot of other things that you can do. I mean, the horizon is huge for the stem cells. We are still wondering what other things we can do with the help of skin cells. They look like this. The skin cells are like this. They are more, have a proper shape. Please zoom this slide more, or you can use the internet to see the difference between the stem cells. The iPSc, they are like less differentiated and less specified structures. So this is possible, and that is exactly the reason. If you can remember, somebody asked me कि China में एक experiment होता है. मैं conclude कर रहा हूँ just 2 minutes में class को. China में experiment होते हैं spinal cord injury में. People are using the stem cells to go for therapy. The answer is yes, it's perfectly on the way. But to use something for a therapy, you need a very strong, approval processes. Every country has its own. The biggest of all, सबका बाप जो है वो FDA है. It is like FDA, which allows a certain therapy or drug molecules to be used for therapy treatment. It requires very strict information and studies. So it took a lot of time. but every country has its own. Salim, you don't need to write your name here. I have explained this thing many times. So मुझे chat में देखना पड़ता है कि शायद कोई question ना हो in the mean. So the therapy, the FDA is ultimate authority to produce drugs and to produce other things. So every country has its own drug regulatory authority. Korea has its own. China has its own drug regulatory authority. They can allow. So China is allowing the stem cell therapy. So in case of spinal cord injury, what is happening? The connection between the neuron is lost because of the injury. These two things cannot connect with each other. The connection is lost between the two things. And we can't push the neurons to grow and connect with each other as well. हम neurons को push भी नहीं कर सकते. So what those scientists are doing there, वो जो laboratories हैं बहुत सारी जो ये काम करती हैं, पाकिस्तान से भी लोग जाते हैं treatment के लिए. But they are using their skin cells or other cells, convert them into induced pluripotent stem cells, and then mix them with the things which can transform the induced pluripotent stem cells into the neurons. And then they inject those things here. So injecting the induced pluripotent stem cells together with the ingredients which can transform them into a neuron. So we are producing new neurons here. So what actually happening? So this is the breakdown thread, one side of the neuron. This is the breakdown thread from the other side, spinal cord mein, and we are generating a stem cell and also giving the conditions. So this stem cell is going to become a neuron. It's trying to differentiate and produce a neuron. It is trying to connect these two ends. Somehow it is trying to connect these two ends. We don't know it is connecting the wires in the right way. ये बहुत अहम बात है. You know, this is a wire called A. This is the wire called, ये neuron का connection है. A should be connected with A. B should connect with B. And there are hundreds of thousands of neurons are out there. So this we are not sure whether we are connecting the right wires, but we are connecting. So after the treatment, we are not sure that the person will start moving in the normal way or not, but the person will start, moving and sensing the lower parts of the body. Of course, the person will need a lifelong therapy treatment to gain the motor control once again. I will explain this thing in more detail, that why it is so complicated. But at the end of the day, the person will be able to feel or to move the things of the lower parts of the body. Of course, at the start, it will not be like this. ये कोई normal movement नहीं होगी कि मैं हाथ move कर रहा हूँ तो कर पा रहा हूँ, ऐसा नहीं होगा. Okay, thank you.